Okay, our next session is led by someone who needs no introduction in dairy genetics, but I'm going to give her one anyway. So Professor Jennie Pryce is one of the most respected figures in dairy genetics as the Research Director at AgVic Research and Professor at La Trobe University. She possesses a deep understanding of livestock producers' needs and excels at applying research insights to other ruminants. She's also just a really great science communicator. So welcome to the stage, Professor Jennie Pryce. Previous slide. To the previous and holding slide, please. Oh dear. Okay, thank you. And it's not just Jennie Pryce. Sorry, AV guys, I should have made that clear. Jennie's actually going to be joined by a stellar panel of researchers from Agriculture Victoria Research who are pushing the boundaries of dairy science, including Dr. Ruidong Xiang, research leader in computational biology with over 80 papers published in top journals, including Nature Genetics and Nature Communications. Dr. Amanda Chamberlain, who has devoted over 25 years to a career in research and is a pioneer in the genomic prediction of health and fertility in Australian dairy cattle. And finally, Dr. Iona MacLeod, a senior research scientist who has a fascinating career spanning both continents and species. Together, they'll give us a look at what's next for DairyBio and the future of herd improvement. So please welcome both Jennie Pryce and her panel discussing what does the future hold for DairyBio. So thank you.
Thank you very much. Thank you, Xiang. It's always like a proud supervisor moment to have, or to see one of your PhD students flourishing so much in their career. So congratulations, Xiang, with what you've achieved so far. And fabulous to have you here as the MC of Herd. Really enjoying your introduction, so thanks. And it's an absolute honour to be sharing the stage with my fellow AgVic researchers, Ruidong, Amanda and Iona. And we always super value coming to Herd because the interactions with farmers and service providers is so important in terms of developing and progressing the sort of research that we do. So really appreciate any conversations that we have and any feedback that you have on the sort of research that we're doing. And I think actually one of the themes of what we're talking about today, and one of the things that makes us so good and so vibrant as an industry, is the way that we work together as a dairy industry to achieve really great results. And I don't think there is a better example of it across our entire dairy industry than what we have all achieved together in herd improvement. And I think this graph demonstrates it so well in terms of the rate of genetic gain that we have seen post-genomic selection and since all the major investment in dairy genetics research. So hopefully you can see the colours in the graph showing dairy future CRC, first of all, in green. And then in the middle, we've got the first DairyBio iteration. And then in the yellow, you can see that we have the most recent DairyBio iteration. And the partnership that we have as an industry has led to this transformation in terms of rapid genetic gain in our bull populations and also our cow populations. And on the back of that, we've seen countless or lots of different breeding values that we've introduced over time that are now available to the dairy industry. And the most recent one being the Sustainability Index that you'll hear about later today. So it is actually, as for me personally, it's very humbling to see this graph, and I love showing it. So thanks to the person who first created it, I think was Leanne Monks. Really, I get a lot out of using this slide in just about every presentation that I do nowadays, just to really make that point in terms of working together and the sort of things that we can achieve. So, without further ado, I'm going to now grill the three sitting nervously on the chairs in the science that we're doing as part of DairyBio. We're going to cover mainly the genomics side of things today, and I hope that you'll find this as interesting as I found it in terms of putting it together. We're going to start with Ruidong first. So, everyone has heard what a SNP is, but could you remind us to start with? Let's talk about SNPs.
All right, thanks very much, Jenny. I guess I'll use this slide as a tool to cast a break and to attract the jades from audiences and the panels. I guess I'll start with one of our major tasks of research is to deliver tools that enable DataGene to calculate better Australian or more reliable Australian ABVs. And one of the most recent tools that we delivered is something called the customized SNP chip, which is enriched of not just SNPs, but cores and mutations. So there's obviously a difference between a SNP and cores and mutations, as this little graph shows on the top left-hand corner, that a cause and mutation usually have biological functions, and they tend to cause the variation in the population of our phenotypes. And they also tend to affect many traits, could affect milk production, fertility, or body condition score, if you will. And they tend to have an effect that are consistent across populations. So for example, they have a similar effect profiles across Holstein and Jersey. And what we have on the SNP chip panel, which are called SNP genetic markers, are just tagging them, which are shown in a graph as gray dots. So they have similar effects, or the smoke, look like the smokes of the gun, which are the mutations, but they're actually not the causal ones. So that's the difference I hope I explained between the SNP markers and causal mutations.
And what do we do with the causal mutations?
So obviously we first want to find them, and that shows in this little flow chart that we first need to have a reference population and developed, and we use various tools to find map them. We first actually need to impute the low density panel to full sequence, so there will be positions for causal mutations, and then we find map them, find them. Once we got the list of the causal mutations, we've got several ways to utilize them. So on the right-hand side of the graph, you can see we first could integrate them into genomic prediction to calculate a better ABV. So we could weigh those SNPs that are causal differently than the ones that are not causal. We could also start the genotyping from a set of, as I said, the customized chip containing causal mutations, so they have better imputation accuracy and maintain the prediction accuracy across populations. And the second pathway for these causal variants are, well, we can look at the causal ones that are lethal or recessive in the population. I think that's something Iona will touch on in the later slides. And also we're looking at the variants affecting population health of the cows. So mastitis or infectious disease, that's something Amanda might touch a bit in the later slides. And there's a dashed arrow pointing from causal mutations to cell line validation. Obviously, they are discovered from computational pipelines, but it's always good to use wet lab approaches to verify their functions. Another potential reason for that interest is maybe down the track a couple of decades later, gene editing might be possible, as currently there are edited animal products entered US food chain, in Japan, and in UK as well. And if that to be happen in the future, and we have the wet lab data to support these functional variants, we might be able to do something and we'll be one step further or more ready than the others. Maybe someday we could look at these dairy calves and using some of the interventional approaches to boost their muscle growth when they are born.
Thank you. Yeah, a lot of, I suppose, insights into what may become our future with knowing those causal variants. Now, somebody who has dedicated their career to genomics and causal variants is Amanda. And I'm going to slip over to this slide now, which talks a lot about the project that you are leading in DairyBio, which is the Transition Cow Health Project. A recent DataGene survey raised the importance of transition cow health, and it's a real priority for dairy farmers. Can you just describe what it means to you and what we're doing in this space?
Yeah, sure. Thanks, Jenny. I don't think I need to explain why transition cow health is important to dairy farmers, to this group of people, but this diagram just highlights that there is variability in the cow's ability to cope with that negative energy balance. I really like this image. It was produced by Tim Luke, which some of you may know, for his PhD project. And so it demonstrates that we've got cows in herds that cope really well with negative energy balance. They reach peak performance, they sail through and have a successful lactation. But there's cows that have subclinical disease, they'll have a dip in their performance, but they'll still go on to finish their lactation, and there are cows that get sick and they don't complete their lactation. So what we're aiming to do is to produce genetic breeding value, similar to your other genetic breeding values, that farmers can use to find those cows that are going to cope well with that negative energy balance. The first step to do that has been to produce a reference population of animals that have health phenotypes, and what we've done is gone out and collected blood samples from over 10,000 cows, and we're measuring important biomarkers that are the kind of gold standard measurement for health records that we've got available to us. We use that as a reference population for this transition cow health phenotype, and we'll produce a genomic breeding value with that. But we're also doing some of the work that Ruidong's just talked about, where we're trying to narrow down which genetic markers are actually causal for a cow who is the difference, I guess, between these blue cows and the black cow, the blue and the red cows you can see on this screen. So we're producing a lot of biological information. So we're looking at the difference between cows in very negative energy balance and in positive energy balance, and biologically what makes those cows different from each other, or those time points different from each other, and cows that cope well, what makes them different to the cows that don't cope so well. And Ruidong's produced some amazing methods for us to be able to use that biological information to actually pinpoint the genetic variants that are most likely to be causing those differences. And we'll feed that into our custom SNP chips that will then produce a more accurate breeding value once we have it.
Cool, thank you. We're going to slip over to this slide now, which is the huge target that you had to get 10,000 blood samples from herds across Victoria into some other states for this project. And in particular, I want to talk about how you've worked closely with farmers and other collaborators in this project to achieve this target and the goal that you set yourselves. And can you give some examples of what you do on farm, for example?
Yeah, sure. It was an ambitious target, I think, and in particular when we were putting this project together, we were in the midst of a global pandemic with restrictions that meant scientists from Melbourne couldn't even leave the metro area. So getting out on farm to collect blood samples was it was a really, really tough thing to start to do. But over the last couple of years, we've been out to 18 different herds and collected I think the number's 11,164 blood samples. We've done that by working very closely with some amazing people, Laura Calder from DataGene, Mitch Crawford from Rochester Vets, and Bernie Baxter from Dairy Australia have provided us with great introductions to some amazing farmers who we must thank for letting us come out onto their farms and hopefully not too much interruption to their usual day. But we did go out, we were there, we had teams of people taking blood samples, processing them on farm. It did mean we were there way longer than the farm staff were, and I hope we remembered to turn all the lights off when we left. But it's been an amazing experience to get out there and see the passion that farmers have for improving the Australian dairy herd and helping us with doing that. We've also got some amazing collaborators. Ian, who you just heard from the Dairy Up program, has provided samples to contribute to this population as well from the New South Wales herd, from some of their experiments, it's just always great to see people keen to collaborate, because this sort of stuff is really hard to do. It's really hard to go out and get these sorts of novel and new ways of measuring health, in this case, from dairy cows. So we collaborate a lot on this project. Obviously, we've collaborated with Ian and his team from New South Wales, but we've got international collaborations as well. And that's because transition cow health is not something that is important to Australian dairy farmers, but it's important to dairy farmers globally. And so we have worked with Lactanet from Canada, University of Cornell in America, and most recently we've struck up a collaboration with a team of researchers from Germany, from 3 different universities over there. And through those collaborations, we get access to samples, we get access to data that is really hard to come by. Getting health records from cows in Australia is really difficult. And we get access to expertise. So we had Jess McCart from Cornell University come and spend 12 months with us last year. And she's a world leader in the epidemiology of transition cow disease. So collaboration is really important when you're trying to put something new together, like a breeding value that hasn't been done before.
And I think this slide speaks volumes in terms of teamwork, just seeing the number of different faces, including Xiang.
Yes.
See if you can spot her.
Xiang was taking blood samples for us while her house was flooding in Rochester in 2022, so an absolutely amazing effort that day.
Thank you. Right, now we're going to change topic slightly and talk about deleterious mutations and diversity and improve herd fitness and genetic gain. So Iona, can you please explain what a deleterious mutation is and why it's important?
Yeah, so deleterious mutations are often called by many names. They're essentially a causal variant that Ruidong and Amanda have been talking about, but they're ones that cause some really harmful aspect to the health of the cow or bull. And you can see in this little cartoon, we've got a carrier bull and a carrier cow. They are completely healthy, and this is what we call a recessive type of harmful mutation. And so the expectation is that if we mate those 2 carriers together, that on average we're going to get 1 in 4 of the offspring will be affected. And this can be seen in different ways. So sometimes it may be that the embryo dies early, and you may not spot that because you just think it's poor fertility, unless you happen to know that you've got 2 carriers that you've mated. Another way it can manifest itself is, for example, the Holstein cholesterol deficiency, or HCD, where the calf is born looking reasonably healthy, but then gets sick, gets scours, etc., but there's nothing you can do to actually get it better, and eventually it just dies. So clearly a lot of economic losses and welfare issues associated with these harmful haplotypes. And in the dairy industry in Australia, maybe Jenny can click on, we've been able to look at the frequency of the carrier rates for a whole range of harmful haplotypes that have been able to be. They've actually been able to find the causal variants because we've run a project called the 1000 Bull Genomes Project, which was a global collaboration. And through that project, by sequencing a whole range of breeds and different animals, it's helped us to be able to identify those harmful mutations. And what you're seeing in the graph on the left-hand side, the bottom left-hand side, is the frequency of carriers in the Australian Holstein cow population, and the graph is showing the cow birth date. So you're seeing the progress of the carrier rate with time. And what's nice to note is that the carrier rate is generally decreasing for most of those variants that we now know about that are causing these bad effects. It's interesting to note there that the much higher line that you're seeing is a new mutation that has been detected, which was found in the French Holstein population by researchers there. And what they detected was that this is having a huge impact on whether a heifer reaches, past their first lactation. So we're currently investigating that in the Australian Holstein population to see if it has the same effect. Sorry, maybe, Jenny, if you could just go back one slide. Yeah, so that graph on the right-hand side there is just simply showing how very quickly something can rise in carrier rate if we're not aware of it being there in the population. And so that example is just the JNS, or the Jersey neuropathy and splayed limbs characteristic. So it got to a very high level. So sorry, Jenny, you can pop on now. So what are we doing in our research? Well, what we're doing is clearly these new the harmful mutations are going to keep arising. So on average, every calf that's born, we expect that it's got 30 new mutations that have never been seen before. And amongst those 30 mutations, occasionally, by bad luck, it may have a really deleterious or harmful impact. And a recent example is the Holstein muscle weakness, which many of you will be aware of, and potentially has a huge carrier rate. This is in the US population. So what are we doing about this? We're researching with a new sequencing technology called long-read sequencing. This is going to complement our 1,000 Bull Genomes project. We've already started this long-read sequencing consortium that we're leading. We've got about 18 different collaborators at the moment, most of them researching dairy. And essentially, that's allowing us to detect mutations that were not possible to detect before using the older sequencing technology that we had. And just the last point I just want to make is reiterating the point about collaboration, how important it is to do this work. We really need good on-farm data. We also need good collaborations, and the diagram on the right-hand side, don't worry about the details, but simply I wanted to draw attention to the fact that in US, this is a new proposal that they come up with to better streamline their system for reporting and managing harmful mutations, where there would be shared governance between the breed societies, the AI companies, and the labs. But on the left-hand side of that diagram, those grey boxes are the research groups involved, and the ones I've circled there in purple are two that we're either leading or collaborating in. And it just brings home the fact that in order to do this, it's a global issue. We use AI bulls globally. And the diagram also reiterates that all of us, that almost everybody in the room, has a part to play in making this research successful and trying to reduce the lag time to detect causal or these harmful mutations. So I'll maybe finish there, Jenny.
Okay, thank you, Iona. Just before we finish up with this session, I want to, I suppose, draw it to a close in terms of we talked mainly about genomics and the use of sequence data. We are going to hear from others throughout the herd conference. So we've got Michelle talking about calves, Laura's going to be talking about heat tolerance, Fazel's going to be talking about sensor information, for example, and Christy's going to be talking about reducing methane emissions. So hopefully between us, we're going to cover the whole breadth of the DairyBio programme. We're harnessing all sorts of different technologies to be able to make use of this data. And I think one of the things that's super interesting and is going to be very powerful for the future is the other AI in terms of using that to improve the way that we do our predictions for both genomics and predicting all sorts of phenotypes. So with that, it's been a great pleasure to present to you, and hopefully collaboration has come through with all of us talking. We really value the input of farmers in the room, especially GINFO farmers who contribute so much of the data that we use. And our partnership with DataGene is obviously super important in terms of bringing these breeding values to you. Thank you very much.
Great, thank you. Thank you so much, Jennie, Ruidong, Amanda and Iona. We are very close to lunch, so instead of taking questions now, I encourage you to memorise these faces and then try and grab onto them during the lunch period. So thank you very much. Have a seat, thank you.